Well, first of all, thank you for having me here. And I'll be talking to you today about an effort that has been going on at the University of Michigan for the last 6 or 7 years. I'll summarize our key findings throughout those years in a little bit of a vignette style, showing how proteomics can be integrated with genomics to bring some insights into cancers. But I would also like to take the opportunity of this talk to introduce you computational people to this brave new world of proteomics. So what I'll end with is really a little bit of a plug why everyone should be interested in proteomics. I was definitely coming into this line of work, let's say, I was very much a skeptic about the value proteomics brings. I think right now it's the next best thing. So I would introduce you to some key problems in proteomics and proteogenomics and hope that as everyone in departments like this got excited about single cell sequencing and various other types of digital sparse data, I hope that some of you will be equally enthused to jump into proteomic data, which, while having very different characteristics, has also very, very substantial computational challenges that just wait to be tackled. So the outline of my talk, so one thing I would like to emphasize, since we are quite a small crowd, please interrupt. Please, if we turn this into a discussion at some point, that's fine. we don't need to go through all of the slides. So if you're interested in something, unclear about something, or just do not agree with something, please interrupt, and I think we'll have a good time. So most of you know, or at least probably this is one thing that you actually should know, is that mutations are necessary for cancer. Cancer doesn't develop without mutations. We can find mutations in all cancers, and with some few very prominent and a little bit elusive examples, essentially every tumor that we sequence has a bunch of driver mutations that contribute to it. You can see it on the graph on the left that the tumor mutational burden varies a lot across cancer types based on their histology, but in general, even the lowest still have hundreds or thousands of mutations in each genome and at least a handful of mutated proteins. We know the sources of those mutations quite well. We know that some of them are environmental, some of them are replicative, and some of them are inherited. And by first approximation, the risk of an organ developing cancer really depends on how the cells within that organ are proliferating. The more proliferative cells there are, and the more frequent they proliferate, the more likely you'll get cancer in your lifetime. This is why things like the colon, which has to constantly turn over and replicate, are associated with the highest risk of developing cancers. And the contribution of those environmental or mutational sources or genetic inherited sources of mutations varies a little bit if we go from early onset cancers, like let's say if you get a cancer in your 40s, that's unlikely unless you're a carrier of a germline predisposition mutation. On the other hand, if you're older, then typically most of your cancers are driven by somatically acquired mutations. And most of the mutations, I would say 99% in an average tumor, they are passenger mutations. They provide no proliferative advantage or no advantage at all to a tumor. Just a small fraction of them actually drive the cancer forward. And for example, for some of them, we know very much what they do. And for some cancers, we even know the order by which the mutations arise to give rise to cancer. So in this very, very influential, one of the key discoveries in cancer period, discovery by Fearon and Vogelstein, we can see how different mutations contribute to the onset and then progression of colorectal cancer. And those mutations do not generally occur randomly. Rather, there is a specific sequence to them. So if we look at that, it appears that we have made quite a big progress. We sequenced essentially every single tumor there is, or at least that we can categorize or acquire samples in sufficient numbers. and through statistical means identified cancer genes. And here, the definition of a cancer gene is very statistical. It's essentially genes that are mutated more frequent than one would expect by chance. And by chance is a loaded word because how we model the null model in such a calculation is difficult, and one can disagree about certain parameters here, but and hence there are many methods. But on average, I would say most people would agree that there are about 1,000 genes for which there is quite good evidence of them being recurrently mutated or altered in cancer. Out of those 1,000 genes, probably half of them have very solid evidence, so solid evidence that they are generally included in testing panels, somatic mutation testing panels, as you may get if you are being treated for cancer. However, out of those 500 or so genes, only 166 are, as of yesterday, actionable. So what does it mean for a gene to be actionable or for a mutation in a gene to be actionable? Well, that means that somebody can do something with that knowledge, right? And in the clinic, there are really not many ways a clinician can actually make use of that information that a certain gene is mutated in a certain tumor. Well, some of the mutations are diagnostic, the 105 mutations here. What does it mean? Well, typically that means that the mutation helps the clinician refine what type of cancer it is or what subtype of cancer it is. Some of the mutations are prognostic, which means if you have that mutation, then you are less likely to live long versus if you don't have it, you might live longer. Certain associations like that. So prognostic information is important, but perhaps not as important. Predictive information, though, is the most vital aspect of that. A predictive mutation or a predictive biomarker means that knowledge of that mutation means that the patient can be given a specific therapy. And in effect, that we can predict whether a patient will or will not benefit from a particular mutation, from a particular therapy based on the presence or absence of that mutation. So if we look at that, only 79 genes out of those 1,000 carry currently such predictive information. And an optimist might say, well, okay, well, there's a long way to go, right? We can expand from those 79 or those 166, and then we have at least 576 that we can still explore for being biomarkers of various types, develop drugs against them, and so on. But in the reality, it's not as simple. One reason it's not simple is because mutations, especially the ones that, well, the majority of those 1,000 are not very recurrent. So recurrence obviously depends on the cancer type because each cancer develops specific mutations. But if we look, for example, at this analysis of 1,000 prostate cancers, you can see that most of those mutations most of the genes with mutations are not very recurrently mutated, many of them below 2%. So the prevalence of those mutations is not large. And for that reason, it may not be actually economically viable to develop therapies or even study them because the cohort sizes required to even enrich for such patients become infeasible. Also, many of those genes, despite being recurrently mutated, are not druggable, at least not druggable by the current mechanisms by which we develop targeted therapies. So some might argue, well, haven't we maybe not discovered yet all of the cancer driver genes? If we sequenced more, maybe we would find some more. And there have been many good studies, essentially power calculations of sorts performed, which try to estimate how much is there left to be discovered. Most of that work has been done in Getty Getz's lab in I guess the last 5 years. And to summarize all of those studies, it doesn't appear we are missing that much. Yes, if we increase the sample sizes by 10-fold, maybe we would discover a handful of genes mutated at a frequency or prevalence of less than 1%. But overall, for most of the cancers, now with hundreds of thousands of tumors sequenced, we seem to have discovered as much as there is in the protein coding genome. So some might argue, well, shouldn't you then sequence the non-coding genome, right? The protein coding genome, it's just 1% or 2% of all of the bases in your genome, maybe there's something to be found in all of those introns, all of those UTRs, and all of those intergenic regions. Well, unfortunately, it turns out that's also not the case. Many people have tried, and I think there's a correlation. The more experienced the lab is with the wielding of the genomic tools, the less they find. So in general, it doesn't appear there's much to be discovered in that space. So that obviously means, well, maybe it's a little bit of a beginning of an end for cancer genomics, because we'll not be discovering many more driver genes, driver mutations. We might still find clinical associations of those that we have discovered so far, but perhaps we are a little bit scraping the bottom of the barrel here. But it turns out genomics is not everything. And there are many examples of that. And I chose to show you only two of those examples to highlight that looking at phenotypes sometimes might be more worthwhile than looking at the genotypes. So in this example, again, going back a little bit to the colorectal cancer model, there are two types of colorectal cancer: microsatellite unstable colorectal cancer, which is a minority of colorectal cancer, and microsatellite stable colorectal cancer. And it turns out that microsatellite unstable tumors respond to immune checkpoint therapy because they carry a lot of frameshift mutations, which generate neoantigens. Those neoantigens are recognized by immune cells, hence stimulating the immune cells through immune checkpoint blockade inhibitors, reactivates the immune cells and has an anti-tumor effect. So based on that very logical, very scientific line of evidence, immune checkpoint blockade is approved by the FDA for all tumors with microsatellite instability. But this study here by Mlecznik, one of the most interesting studies, asked the question, well, do we actually need to know the fact whether a colorectal cancer is microsatellite stable or unstable? So what they did is they developed a phenotypic score. And this was a phenotypic score done by pathologists by looking under the microscope at the tumor sample, counting T cells, looking where they are. It's actually a quite complex scoring scheme. But once they developed the scoring scheme, they put it into this Cox multivariate model. And what you can see is that the genotype doesn't matter anymore. Whether a sample is microsatellite unstable or microsatellite stable is irrelevant. What matters is whether you have those T cells inside it. So the phenotype, in a way, outperforms the genotype in predicting responses to immunotherapy in that cancer. Another example is the recent rise of ADCs, antibody-drug conjugates. So rarely in cancer do we really see big breakthroughs in therapy that affect a lot of people. But the last 2 years, I think, brought two such breakthroughs. The first breakthrough was the approval of a therapy, which is an antibody-drug conjugate in metastatic second-line breast cancer. So that's a pretty advanced cancer, right? The cancer was metastatic, was treated with one round of therapy, and then this is the second line of therapy. And in that setting, this antibody-drug conjugate, which recognizes the presence of sometimes low amounts of HER2 on the surface of those cells, this is not a genetic classification. These cells are not HER2 amplified. They are just have some HER2 on them, it improves over the physician's choice significantly in both disease-specific survival and progression-free survival. And similarly, just this week, the results of a trial read out where a bladder cancer, first-line metastatic bladder cancer, was treated with a very similar antibody-drug conjugate, which targets Nectin-4, again, a protein that is expressed on the surface of bladder cancer in the metastatic setting as a first-line therapy. Why this is important? Because we haven't seen really advances in the treatment of first-line bladder cancer for 30 years. So targeting the phenotypes sometimes actually gives meaningful benefit to patients. So where we are with respect to that. So at the University of Michigan, we are one of so-called Proteogenomic data analysis centers. This is the computer here on the left, which is a center which is part of the Clinical Proteomic Tumor Analysis Consortium, CPTAC. And our role as this Proteogenomic Data Analysis Center is really to support this whole effort in data analysis capabilities. As you can see, this is quite complex of an organizational chart. There are many people involved. This is more than, I think, 15 institutions play some role. Some of those institutions are tissue source sites. Some of the institutions perform the proteomics, and 3 institutions, the Broad, Baylor, and U of M, are responsible for the genomic data analysis. So here, I would like to really, before really showing our work, I would like to acknowledge all of the people that have contributed to it, at least the people that have contributed to the results that I'll be showing, but it's not only my lab. I'll be showing the results from Noshat and Alec, who are bioinformatics and pathology PhD students, but also from Alexei's lab, his 2 postdocs, and Leo, a bioinformatics student, have also performed many of the analysis. The Chennai lab, where Mohan, Chandan, and Rahul, a pathologist, have also greatly contributed. Gil, who supervises all of us and every week joins us for a 3-hour Thursday afternoon data analysis meetings, and many CPTAC collaborators. So it's really a group effort. I'll be showing many more results than one lab or perhaps one person could feasibly accomplish. So what are we doing there? Well, we have 2 sets of data analysis pipelines. The first pipeline developed in my lab, we called TPO for Turnkey Precision Oncology, is a pipeline, I should say many pipelines, but under one umbrella of algorithms and workflows to analyze genomic data. I'll not go into the details of it, but it's really as comprehensive as it gets and as tuned to the types of data CPTAC is producing. On the other hand, we have the proteomics pipelines. The pipeline is called FragPipe. It has been developed in Alexei's lab for, I think, the last 10, maybe even more years. It's also an umbrella term for many algorithms. It also runs on the cloud and really powers most of the proteomic analysis that really make CPTAC feasible. So CPTAC generates lots of data, mostly genomic sequencing, transcriptomic sequencing, DNA methylation, proteomic, shotgun mass spectrometry, metabolomics, DNA methylation, all of these types of information on a generally large cohort sets of tumors. And our role with all of that data is to derive some meaning out of that and answer generally clinical questions. And the questions, they vary. Obviously, each cancer type is different and the clinical problems to be addressed within each cancer vary. But there's a framework of how we approach it. And I guess there are 2 really ideas that I would like to highlight here. The first is that we try to generalize. We generally start with deep analysis of individual tumors, like the one depicted here, and we try to generalize those findings to as large of cohorts of patients as possible. Why? Well, because while personalized medicine is a good idea in general, the treatments that are available or should be applied to groups of patients. It's simply impossible to attune everything to the completely unique molecular profile of a patient. And we also want to generalize the data or the molecular finding at the molecular level, going from individual mutations in driver genes all the way to signaling networks. Again, trying to see what is common rather than what is completely unique. And ideally, what we want to do is we want to make novel observations that are predictive in nature, because that's what we currently lack most. We do not often have the biomarkers and the clinical tools to determine who will respond to therapies, who will benefit, and what types of therapies they should be getting. So lots of our effort is on trying to identify predictive biomarkers. And I prepared 4 vignettes of various clinical questions and various molecular analysis that we have performed. And I think, looking at the clock, I'll skip the first one and jump straight into the second one, to not really run out of time. If we have time left, we might want to go back. So the second study that I want to highlight here is ovarian carcinoma. Ovarian carcinoma is a very deadly cancer. The biggest problem in ovarian cancer is by far late diagnosis, because it's diagnosed in most patients with vague symptoms at stage 4, which is already metastatic ovarian cancer. The survival, the 5-year survival for ovarian carcinoma is minuscule. So the biggest problem there is really diagnosis. So one problem that should be addressed is definitely diagnostic biomarkers. The second problem in ovarian cancer is that although most patients respond to platinum-based therapy, the response rate is about 90%, approximately 10% do not respond at all to platinum-based therapy. This is a problem because how late those patients are diagnosed, there typically is no second option. So once the patients are given platinum and wait the 2 or 3 months to see whether they respond or not to that therapy, it's typically too late for them to be enrolled in a second, for example, in a study, or for example, be given a different therapy. So we are delaying useful therapy for those 10% of patients. And as part of CPTAC, we tried to address the second problem, essentially trying to predict which patients are platinum refractory. This is the group of patients, one could call them exceptional non-responders. And this is because unlike the majority, which very well respond to platinum and are called platinum sensitive, these patients do not respond at all. And there has been at least 30 to 40 years of research on trying to find a predictive biomarker of platinum response, but none of those efforts panned out. One of the PIs on this grant called it the biomarker cycle of insanity. And she argued, Amanda, that the reason for that is that everybody all the time focuses just on one biomarker, tries to validate them, and nobody really looks at all of them at once and really tries to pick the best combination of proteins or genes or transcripts that could help us. So to address this question, they developed a very interesting cohort, essentially enriching for the refractory non-responders, exceptional non-responders, and contrasting them to the standard sensitive high-grade serous ovarian cancers. When I say sensitive, that doesn't mean that those patients are cured. Those patients respond, derive benefit from platinum therapy, but then typically within 6 to 12 months, the tumor comes back and patients ultimately die shortly after. So it's not a comparison of cured versus not cured, but rather it's a comparison of initially sensitive from initially refractory disease. So this cohort, I should add, it's very unique because it has been sourced from 3 different institutions from pathological archive. That means that most of the tissues, the specimens that were used for this molecular profiling were actually FFP, formalin fixed paraffin embedded. This is a standard way by which hospitals save tissues for storage, for archive, but it's not the best way to preserve either nucleic acid or proteins. So in many ways, technologically, this was a very difficult feat to get both high-quality genomic information as well as high-quality proteomic and phosphoproteomic information out of that. So when we had the first look at the data, actually, the results were not that encouraging. If you look on the left, this is a plot which shows the fold change of sensitive to refractory, essentially trying to find whether any individual proteins can help us predict sensitivity or refractoriness to platinum therapy. As you can see, only 3 proteins here are highlighted, and these are the only 3 proteins which marginally were validated in validation cohorts. This was not enough to build a robust biomarker. We also looked obviously at the genomic data, tallying all of the mutations, and seeing whether certain mutations are enriched in one group versus another. And also, unsurprisingly, we found no difference. So what does one do running into such a snafu? Well, we went in essentially two different routes, and I'll talk only about one route. But the first route, this was effectively the planned route as outlined in kind of design of the study, was to apply more high throughput or more machine learning based approaches rather than relying on single biomarkers. This work was mostly done in Pei Wang's lab at Mount Sinai. What they did is based on literature curation of finding previously genes and proteins that could have been associated with platinum sensitivity, also relying on mouse models of platinum resistance, acquired platinum resistance. They nominated a bunch of pathways, but also individual genes that had a very high likelihood of being associated with refractoriness, built machine learning models, and based on the output of those models, tried to predict whether a patient will or will not benefit from platinum. And as you can see from the ROC curves, they achieved quite a good AUC. And these are the three validation cohorts. In all three independent validation cohorts, they could still see the signal. And currently, this biomarker, which is a machine learning biomarker, is being evaluated on a clinical trial. What we did, however, is we tried to do a similar type of mining for signal, trying to understand whether we can use the information of chromosomal instability between sensitive and refractory tumors to identify those that respond versus those that do not respond. And I should add that high-grade serous ovarian cancer is among the most genetically unstable tumors. Well, perhaps it's even the most unstable genetic tumors. And you can see it here. I'll use those plots repeatedly, so I'll explain them once, but if you're confused, please ask. Each row here corresponds to a patient. So we have approximately 150 patients here, 150 rows, and each column corresponds to a locus at a certain resolution. And the loci are arranged by chromosomes. So this chromosome 1, chromosome 2, chromosome 3. So for example, you can see that most of those tumors have a gain on chromosome 3. And we know that it's a gain because it's color-coded in red. Here you can see that copy number greater than 4 is dark red, copy number 3 is light red, and so on. The blues, for example, the losses of chromosome 4, you can see them also. So this is a way to just see. And if you were to look at it, obviously, both groups are very, very unstable. The sensitive group here in blue, and the refractory group here in blue, and the sensitive group here in yellow, they do not seem to differ in terms of their responses. But I would like to highlight one observation that we made a little bit by chance, by looking at this data from a slightly different perspective and by organizing this data through the lens of proteomics. So we applied proteomic clustering to all of those samples, and we looked at a set of 5 clusters, trying to see whether those phenotypic clusters of high-grade serous ovarian cancer have distinct genetic patterns. And again, here it seems that all of those groups are the same, at least if you look at it by eye. And here I also introduce another color. So in the previous plot, I showed you that red means gain, blue means loss. So what does green mean? Well, green means copy-neutral LOH. These are generally regions of the genome which do not have a loss, but are also not heterozygous. So they have a loss of heterozygosity. And there are multiple mechanisms that can give rise to loss of heterozygosity. But essentially, in such a region, both chromosomes or both genes or both loci are from the same parent of origin. So for example, both are paternal alleles, both are maternal alleles, but never heterozygous. So you can see in high-grade serous ovarian cancer in general, there's lots of heterozygosity, lots of that we can see a lot of green. So when we organized the data by those clusters and looked at the patterns of heterozygosity, we observed something we didn't expect. When we looked at chromosome 17, we found, as expected, that most of the samples had a lot of LOH, loss of heterozygosity, across all of the clusters. This is expected. And the reason why this is expected is because in high-grade serous ovarian cancer, LOH is nearly universal, and LOH is associated with multiple genes, 4 exactly, that are recurrently mutated in high-grade serous ovarian cancer. But what we observed here was this cluster number 5, which somehow was enriched for samples that didn't have LOH and in fact had a gain of chromosome 17. So that was curious and unexpected. So then what Naushad did is he decided to check whether actually chromosome 17 LOH was associated with response to platinum. A seemingly obvious analysis that, to our knowledge, has not been tried before, perhaps for one reason of not having a sufficiently powered cohort to determine sensitivity and refractory status. And here you can see that after adjusting for multiple hypothesis testing, only chromosome 17 LOH is significantly associated with sensitivity and refractoriness. And you can see it also here. Most of the tumors have chromosome 17 LOH as expected. A small proportion of them are not. And among those that are not, there's a preponderance of tumors that are refractory. So now the question is, well, but it's just LOH. How can you associate LOH with responses to platinum therapy? Again, it comes down to the genes that are on chromosome 17. I'll not attempt to summarize all of the analysis, but just to give you kind of a flavor of how we try to unravel it. First, sensitive tumors are much more genetically unstable than refractory tumors. And one thing that enables tumors to be unstable is having defective p53. And in fact, if we compare tumors that do not have chromosome 17 LOH versus those that do have chromosome 17 LOH, we see that tumors without chromosome 17 LOH have wild-type p53 activity, whereas tumors that do have chromosome 17 LOH do not have wild-type p53 activity. If we look at mutant p53 activity, which is the activity typically associated to missense p53 mutations, we can see the converse. Tumors without chromosome 17 LOH do not have mutant p53 activity or have less mutant p53 activity compared to tumors with chromosome 17 LOH. That is not to say that tumors without chromosome 17 LOH do not have p53 mutations. They still have a substantial number of p53 mutations, but rather those mutations are monoallelic. Only one of the alleles of p53 is mutant. So we were able to propose a model wherein p53 mutations in high-grade serous ovarian cancer can be classified as biallelic, monoallelic, and wild type. If it's wild type, then there's no high-grade serous ovarian cancer, essentially. However, if the mutations are biallelic or monoallelic, the ones with monoallelic p53 mutations tend to be refractory. Okay, so jumping to another cancer and a completely different question, we looked at lung adenocarcinoma. Lung adenocarcinoma, very, very different from ovarian cancer, also sometimes diagnosed late, but generally it's the cancer for which we have a lot of therapies. However, we have some challenges in prognosticating who, not only who will benefit from those therapies, generally the therapies administered based on mutations, but independent of mutations, we lack good diagnostic and prognostic tools. And one thing that was very curious in the literature and in lung cancer research was the fact that there's quite a range of how unstable lung cancer genomes are across the individual cancers. Some of the genomes are very stable, as you can see here. This is a stable lung adenocarcinoma. You can see that it has a very stable baseline of 2 copies with few chromosome arms but generally whole arms or whole chromosomes gained or lost. So such a tumor would be classified as stable because not many chromosomes deviate from the baseline of 2. And there are even more stable tumors. On the other hand, there are tumors which look like this. You can see that there are many more gains and losses. Also, you can see everything is red, which means that there was a whole genome duplication in that tumor. So this pattern of differences in genomic instability was curious, and it was observed before, for example, here in this remarkable study by Teresa Landi, which classified lung adenocarcinomas into those stable ones, those unstable ones, and some intermediate ones. The problem with this classification, though, was that it served really no purpose, because it didn't allow us to predict any particular behavior of those cancers, and especially didn't predict either responses to therapy or survival. We repeated this analysis, essentially quantifying genomic instability, as was used by that study, and what is also the standard way of characterizing genomic instability. And you can see that whether you're stable or you're unstable, your survival, at least in this cohort, but in many previous cohorts, is the same. So here, Naushad thought a little bit out of the box and applied a different way of visualizing the data, which brought us quite a lot of insights. Rather than focusing on the deviation of copy number relative to a baseline, he looked at the degree to which the genome is shattered into pieces. So he looked at segment length, and he compared the density of the segment length distribution. This is not a commonly prepared plot, perhaps to our benefit, but what it shows that there are some tumors which have the majority of segments long, and then there are some tumors which have the majority of segments very short. You can see it also here on this density plot. There are 3 cases here. One case, you can see the red case, it has the majority of segments very long, and contrast it with the green case, which has the majority of segments very short. So we decided to look at the segment length distribution as a way of risk stratifying lung adenocarcinomas. What Naushad did here is he looked a little bit into an old literature of studying spatial processes, in effect, looking at clustering patterns. You can see, for example, on these 2 diagrams here, is that, well, first of all, the length of the segments is determined by the number of breakpoints. The more breakpoints you have, obviously, the shorter the segment. That's logical. But there's also another way of altering the length of segments, their distribution. You can cluster many breakpoints close to each other, thereby leaving room for the long segments to arise. So essentially, given the same number of breakpoints, if there's more clustering, there's higher contiguity. And the statistical theory here allowed us to quantify the degree of clustering of points on a line, such as, and the theory also generalizes to more than one dimension. So in contrast, the genome instability index measures the deviation of copy number variance relative to a baseline, whereas what we call, we've changed the name many times, but we now call the genome contiguity index, it measures segment length and distribution. What's the benefit of doing it? Well, if we look at segmentation of the genome, it becomes clear that actually tumors in which the breakpoints are not clustered, meaning that shatter the genome quite uniformly and generate many segments of short size, rather than few segments of large size and many segments of tiny size, that those genomes tend to do worse. And you can see here that both genomes that are very much shattered, as well as genomes that are not very much shattered, but also not clustered, that these have worse outcomes than tumors that have few breaks or many breaks, but clustered. So this allows us to look at the instability of the cancer genome from a different perspective. We then look at what genomic features are associated with chromosomal instability. Again, unsurprisingly, we found that P53 was significantly associated with instability, mutations in P53, but also TERT amplification. TERT is responsible for DNA maintenance through a mechanism called alternative lengthening of telomeres, and it's essential in some cancers such as melanoma, but also, as appears here, in unstable lung adenocarcinoma. MYC is a very, it's a transcription factor that is necessary for high rates of proliferation. So we found significant associations between those known oncogenes in those unstable tumors. But we wanted to also understand what's going on there from the proteomic side. So having access to the proteomic information for those specimens, this allowed us to compare the unstable to the stable tumors, and we identified IGF-2BP3 and IGF-2BP1, two related RNA binding proteins with very unclear functions as being most associated in those unstable tumors. We chose this as a potential biomarker due to two facts. First, there was a very, very clear association between IGF-2BP3 expression and TERT expression, really bolstering the case that both are present in the subset of unstable tumors. But also, unrelated findings in a different thyroid cancer have shown that IGF-2BP3 is an oncogene in its own right. Recurrent translocation between THADA and the promoter or enhancer of IGF-2BP3 result in massive upregulation of IGF-2BP3, driving a subset of those thyroid tumors. So IGF-2 being an oncogene in its own right made it especially interesting. And here, I'll not go into details, but Rahul, a pathologist in a Rolls lab, validated IGF-2BP3 IHC as a biomarker for lung adenocarcinoma, allowing us to distinguish through IHC between the unstable, stable, and intermediate tumors, and through scoring, was able to show that it's a very specific biomarker absent from the NATs, which are normal adjacent tumor samples, essentially normal healthy lung tissue, whereas it increases in metastasis and is even higher in primary squamous tumors. So a good biomarker picking up the most high-risk type of lung cancer. Going on to our latest work, again, a different cancer type and different clinical questions. We decided to look into AML. So AML stands for acute myeloid leukemia. And I have to admit that before starting this project about a year ago, I knew very, very little about hematological malignancies and AML in particular. And I always thought that, well, compared to solid tumors, leukemias are simple. They typically have a clear driver, a translocation, or a mutation, are also curable in many cases. And I thought, well, that should be simple. Well, nothing further from the truth. AML is among the most genetically complex diseases. This is here a classification of AML based on genetics. This is a paper that came out, which proposed a revised classification based on WHO 2016 classification. And this is already outdated because there is already a new WHO classification. So lots of genetic events allow us to put AML into different molecular buckets. The second thing that makes AML unique is because unlike lung adenocarcinoma, which generally is driven by a few mutually exclusive drivers, so patients with an EGFR mutation rarely have an KRAS mutation. In AML, the patterns are much less clear. Many of the mutations are not mutually exclusive. That means that each individual case is essentially unique in its presence or absence of various mutations in one of the many genes that are recurrently mutated in AML. But perhaps most confusingly and most difficulty to deal with that is that although those mutations are not mutually exclusive, they're also not random. So certain pairs of mutations are more common than others, certain pairs of mutations are less common than others. So there's lots of structure in that even only genetic data. On top of that genetic heterogeneity, there comes phenotypic heterogeneity. So AML arises from mutations in a hematopoietic stem cell, but depending which particular stage in the hematopoiesis the mutation occurred in, and perhaps on other properties, phenotypically, the AML has various patterns of differentiation. So there can be AML, which is fully de-differentiated, or there can be AML, which looks, for example, like a monocyte. And this phenotypic heterogeneity, again, contributes to the already high genetic heterogeneity of the disease. So here, we performed, again, the same type of exercise of gathering lots of molecular data, proteomic data, genomic data, metabolomic data, and using this data to understand the disease a little bit better. And for this particular cohort, we decided to do it perhaps a little bit differently than in other studies. And we decided to see how much can we gain by combining the analysis of RNA sequencing and shotgun mass spectrometry together in trying to classify AMLs. And I'll not go into all of those analyses. This took 3 people about approximately 3 months to really nail down how, first, what computational approach to take, second, how to validate it, and then third, how to derive meaning of the clustering, but essentially using an algorithm called similarity network fusion, which is a graph theoretical algorithm, which in my mind overcomes some of the challenges of doing multi-omic clustering. We arrived at 8 subsets of AML, and I'll show you some of the results and analysis of those subtypes. So the AML8, as we refer to them, are 8 groups of AMLs, which within each group, the patients or the tumors are very similar to each other, and they are rather dissimilar to the other patients. What is remarkable about this classification and really provides me with a lot of joy to look at is how strongly those molecular phenotypes are driven by the underlying genotypes. So for example, you can see that clusters 1, 2, and 3 effectively harbor all of the cases with mutations in NPM1. NPM1 is one of the class-defining genes that is recurrently mutated and used to define a genetic subset of AML. On the other hand, for example, we see that in cluster number 2, we have a high number of FLT3 ITDs. FLT3 ITDs are not used to define a genetic subset of AML, but previously were thought to be rather disparate in their molecular and clinical behavior. But we can see that the joint presence of NPM1 mutations and FLT3 ITDs is what genetically defines cluster number 2. The other clusters all have such distinct characteristics. For example, cluster number 3 has biallelic CBPA mutations. Cluster number 8 is enriched for the RUNX1, RUNX1T1 translocation, and so on. And you can see the various associations here on the right side. In essence, every cluster is enriched or depleted of certain mutations, and every mutation is enriched or depleted in at least some clusters, really showing how genetics determines AML molecular phenotypes. And I'll only highlight one of those phenotypes, cluster number 2. Cluster number 2, we found to be enriched in mitochondrial proteins. Interestingly, this can only be detected using proteomics. We found that cluster number 2 has a hallmark of oxidative phosphorylation, with all of the genes involved in the electron transfer chain being upregulated. And taking advantage of the metabolic data, we can see that cluster number 2 has higher concentrations of ATP and lower concentration of glucose or phosphoglucose. So essentially showing how those tumors rely on oxidative phosphorylation to turn glucose into ATP. We could also predict for those tumors which of them will respond to therapies. We leveraged an external data set called BetaML, for which xenografts were derived and the drug screen was performed, and also proteomics was performed to match up the results from our clustering to the results of that screen. And again, we could identify which clusters or patients with which molecular profiles are more or less likely to respond to which drugs. Ultimately, this allowed us to piece together this matrix, and this is still a work in progress, where each cluster is given a particular profile of mutations, differentiation, sensitivity drugs, and also various other molecular characteristics. Essentially, the complexity, the genetic and the phenotypic complexity of AML is reduced and organized through this analysis of multimodal genomic data. With that, I would like to end this portion and really just briefly tell you why you perhaps could be interested in proteomics. I think unlike genomics, where I think one could argue there's a little bit of a stagnation in terms of both methodologies and novelty, how proteomics really pushes the barrier of what is feasible and what can be actually measured. And I would like to highlight in the next slides what I think are the most promising developments of proteomics, and also perhaps the developments that require most of computational skills. This is not right. So first thing, what is truly emerging right now is protein sequencing. And many of you might think, well, that's a gimmick. Well, it doesn't work yet. And I think that's right. It doesn't work yet, but there have been credible demonstrations that by using enzymatic protein unfolding, it is possible to determine the sequence of a protein by pushing it through a nanopore. I think this will really revolutionize how we approach studying proteomes, because currently our way of dealing with proteins through mass spectrometry is really to match spectra of short peptides to a database. Once we sequence, we don't need to do the matching. We can actually learn the sequence of the peptides directly. The second thing that is emerging and will undoubtedly have a huge impact is single-cell proteomics. And again, you think, well, why do we need single-cell proteomics if we have single-cell transcriptomics, which works remarkably robustly? And I think the argument that should be made here is that single-cell transcriptomics perhaps tells us a little bit too much. I'm sure you've seen many studies that applied some algorithm to some single-cell RNA-seq data and arrived at many clusters and called them cell states. And I think there's quite some argument to be made that maybe those states actually do not exist at the protein level. Proteins in general can be much more stable than RNAs. So maybe what we see as cell states doesn't live long enough. It doesn't reflect itself in the protein definition of a cell, because it's much more stable and essentially smoothened out. So once we have those single-cell proteomes, I wonder how many of those findings of single-cell transcriptomics will have to be revised or at least reinterpreted. The second thing is that measurements, that proteomic measurements, unlike digital measurements such as single-cell transcriptomics or transcriptomics, are much more complex. These are really biophysical analog machines relying on a bunch of physical processes such as column separation, ionization, and so on. And there is a rich measurement theory which goes into developing the right way of deriving values from those measurements. Something I find very exciting, and also something that Alexei's lab is really experienced in, is how to take those complex biophysical measurements and turn them into numeric values that can be interpreted, but lots of work there to be done, especially on the aggregation level. We measure currently through mass spec proteins at the level of peptides, but most of the interpretation we do is at the protein level. And there's a step of aggregation, how we integrate signals from multiple peptides into a single protein measurement. And I think there's lots of work that needs to be done there. First, finally, I would like to highlight more perhaps advanced analysis of proteomics data. You've undoubtedly, many of you tried different types of data integration, RNA with a TAC-seq and so on, various digital measurements. And similarly, we are performing the same thing, and you've seen some of the examples on RNA and protein. But unfortunately, we still do it in a quite dumb way. In a way, we treat those matrices as essentially matrices of numbers. We do methods like regression, factorization, or mediation analysis, but really we just treat those matrices as matrices of numbers. For whatever it's worth, it could be anything. There's no understanding in our analysis that the green matrix corresponds to RNA and the red matrix corresponds to protein, and there's a certain causal relationship between them. If you I'm pretty sure you noticed how big of an impact RNA velocity analysis made, because it proposed the integration of pre-mRNA sequencing with mRNA sequence in a more model-based way. And I think the same first principles thinking can be applied to proteomics, such that we can actually integrate the data in meaningful ways. And if you're interested in that, I would encourage you to look up what currently people understand. If they say a protein QTL or pQTL, and I think you'll be astonished how primitive that definition is. Finally, I would like to highlight that you probably noticed how pangenomics has made a tremendous impact on the genomic community. Now we can represent variation across many species or multiple humans in one graph to represent the human reference genome. And I think we are still quite a bit away from it, but this type of pan-proteomics, really representing variation in our proteogenomic analysis, is not currently possible. There have been many attempts, many poor attempts. I think algorithmic development, similar to these heroic efforts that went into making actually graphs practically useful for genomic analysis, are needed in proteomics that could make a similar impact. And finally, I should mention that you probably heard about the histone code, how different PTMs, post-translational modification on histone tails, dictate the logic of what is active chromatin, what is silenced chromatin. They are deposited in certain order and have some regulatory logic behind that. And we have not yet developed methods which would allow us to get such regulatory logic from all of the different PTMs that we can expertly measure. And here again, a plug for Alexei's lab, who develops the computational method, which really allow us to position and quantify all of the PTMs. But we need the next step. From all of that knowledge of the PTMs, we need to actually find what do they mean, how do they interact, and how to interpret them in the context of biology. Thank you so much.
Okay. Questions? We'd like to start. Morgan.
Yeah, so I'll be the running. Cancer has genetic and environmental factors, and we know sometimes the environmental factor change the pattern of genes involved. You said earlier on there were some genes that were very common, and then a long tail of lots of genes that were involved. So if you take, for example, lung cancer, there are different genes, as far as I understand, involved when you have smoking versus non-smoking, which is sort of my paradigm of environment changes, which genes are involved. So have you observed other cases where, dietary changes that people have done over the years have actually changed the pattern of genes? And do you think we will see a whole new pattern, or you think most of the cancer genes will stay the same?
Yeah, that's a good question. And unfortunately, off the top of my head, I cannot come up with a I cannot come up with a good example, but yeah, that's well taken. I mean, there are examples like cholangiocarcinoma or gallbladder cancer that, depending on whether you're in Asia and you have some infections, it looks completely different. But yeah, like more dietary, I think it's a little bit more difficult. That's an infection by yeah. That's a pathogen infection, yeah. But yeah, that's a good question. Maybe we'll find very specific environmental causes. Yeah, yeah. Yeah, I mean, in lung cancer, you mentioned, right, that there was that big study from Charles Swanton's lab that really showed that certain cancers are associated with pollution, but not smoking. And they have different molecular profiles than the one associated with smoking. But this is also the Asian, non-Asian divide, right? Where non-smoking females in Asia have a very different type of mutational profile.
From the entire CPTAC understanding is that you have RNA as well as proteomics data. And from the papers that I've seen, there's always this scatter plot in which you're correlating RNA expression, proteomic expression. I wonder if there's any work done or if somebody has taken this initiative of sort of coming up with the intermediate value, which defines both proteomic and RNA-seq expression.
Well, we tried to integrate those data sets, but I'm not sure if you can take an average of such disparate values, right?
So you mentioned mitochondrial proteins being mutated in some cases, and it's my understanding that there are nuclear encoded proteins. Are there any examples of mitochondrial encoded proteins being mutated?
Yes. Mitochondrial mutations have been studied. I think the main set of papers came out like 3 or 4 years ago. So yes, there are mitochondrial mutations, and they're extremely difficult to interpret, right? Because there are many mitochondria per cell, and not all of them are mutated. So what is the proportion of mitochondria per cell that need to be mutated in order for it to have an effect? So yes, in clonally expanded cells, we can see some of them having mitochondrial mutations, but I think the next step was very hard to make. What does it really mean? And no clear clinical associations have been made. So they are not routinely being detected or studied, or I should say, recorded as part of kind of the clinical workup of a patient.
One of the features across CPTAC is look for pan-cancer correlations. Are there significant similarities by histology, that's squamous in different organs, adeno in different organs, or any other example you would highlight?
Yeah, it's.
Of course, it includes susceptibility to immunotherapy.
It's a very challenging concept. In general, I think that the concept of pan-cancer analysis is a little bit flawed. And the reason is because cancers are very, very different. And they are different at the individual level, which is perhaps not helpful, but then they are different between groups of them. Histology helps us organize this knowledge of cancer. So there are very, very few things that actually hold across many cancers. I think in my lab, one thing that we focus on is the MHC, which is perhaps one of the few things that actually holds across all of the cancers, because all of the cancers interact with the same immune system. But the pancreatic tumor and the lung cancer and the leukemia, they're also different, that their molecular aspects are very, very dissimilar. The other thing that perhaps holds across all cancers is the aspect of genomic instability and p53 in maintaining the diploid status of cancers. So I think genomic instability as a pan-cancer phenomenon being associated with advanced disease is also quite universal, but how it affects different tumors also differs. So for example, the patterns that I showed about sensitivity, about certain patterns of instability being associated with outcomes in lung cancer, they'll not hold in kidney cancer, and they are unlikely to hold in ovarian cancer. So every cancer still requires every cancer type requires quite a unique perspective and really interrogating the molecular data specifically for it.
Okay. Thank you for a wonderful coverage of events. Here.